piece of cake, no big worries. Secondly, walk out here tonight, well thinking how much time did I use to prepare for that exam? Did I do, John, minimum 20? If you didn't, if you did less than that, like maybe 10, then maybe that's sort of why you got the score that you were getting. We made lots of things. Bottom line is, it does not indicate what your grade will be, but only how much work you and I have to do to help you get an A in this class. There's still plenty of time to make a really good, strong, high, almost 4.0 in this class. All right, part 2, hearts. These are cool and fun because there's no chemicals. You can actually eat these things. They were that fresh when they came out of the animal when I first got them. What I'm going to have you guys do is I'm going to open up the bag. You're going to work in groups of 3 or 4, in other words, per row of tables, right? And it'll be an individual group. You're going to grab a cafeteria tray. You're going to grab a heart out of here.
Oh, it's a bunch of hearts.
All right. You need kindergarten scissors? The guys like they did. All right, let's try some actual scissors here. There we go, a little better. And what you will do, because as you'll notice, there's lots of fresh natural fluid in here, aka blood. I'm going to grab one, just kind of like this. That's I grab 1 or 2. There's 2 here. All right, they're quite large. All right. First thing is this. I'm going to kind of just take my hands and just kind of lightly, squeeze any excess fluid out over here. All right.
Yeah.
Now, to make it easier for you and your lab mate, you're going to come over to your little table, all right, your little kitchen, and you're going to rinse the heart off. Be careful. Everybody pay attention, everybody look and watch. Put your laptops down, all right? When you work with this, all right, it is a high pressure hose. Don't crank it because the water will dave. The water will hit and spray up and outwards, and all the fluids from the earlier lab are now flying through the air, right? So you slowly open it up just a little bit. Rinse the outside of the heart off, and if you really want, kind of cool, put some blood in, if you can find the superior vena cava,
Some water.
turn the water flow on, and watch it come out maybe the pulmonary trunk, because that's where it's going to have to go, right? You're just looking at clear blood now, aka water. Once you rinse the heart off, grab about a good 2 feet of paper towels, right, like this, and blot the heart dry. All right, so it's not just dripping water all over the place. Now, these tissues, these hearts have no chemicals in them. All right, so none of this is going to go in the trash can that I put a step stool on top of to make it harder to get into. Okay, this is going to go in this trash can right here. You're going to take all your dirty mess and you're going to put it right in there. That gets emptied every single night. Okay, when you don't do that and you put it outside here, it'll sit there for 2 or 3 or 4 days, and then you might have noticed maybe during the summer or last quarter there's kind of a dead, rotten animal smell. That was due to somebody putting fresh tissue in the trash can, and they don't empty them every single night. And once in a while, though, it is actually a squirrel that got trapped in there that didn't survive. But usually it's you guys putting tissue in there that didn't work. Now you gotta think about what it is you've been practicing doing, which is hopefully learning the various parts of the heart. And most important is which part of this mass is the anterior side. You remember how to identify the anterior aspect of the heart? Okay, besides saying yes, tell me what you actually would do. What do you do? So you're looking for the pulmonary trunk to come right off of the front of the heart. All right? So that's, I'm looking at this thing going pulmonary trunk. Oh, cool. Look at this nice big thing right there. All right? It's about as big as your thumb. That's the pulmonary trunk. Now I can orient myself and realize this is going to be the right side. I'm sorry, the right side to the left side. I'm backwards. I can see the interior world exactly. Welcome to my world. All right. But what is that serous membrane that covers the heart? What's the name of the sac overall? Pericardial sac, right?
Yeah.
Aren't there 2 layers to the pericardial sac? What are the names of those 2 layers? Parietal, visceral.
And parietal.
What am I literally touching right here? What name would you give what I'm touching? Or? Either one, right? This is either the upper layer of the heart, if I'm asking for heart tissue, or it's the inner lining of the serous membrane of the pericardial sac that actually is the outer membrane of the heart itself, right? It's got two names for the same thing. For example, what is that skin right there called?
Epidermis.
Epidermis. Could I call it something else? How about stratum corneum? Right? Depending upon my is it my integument system or the layers of epidermis, right? Same thing with this. So I want you to find the pericardial sac if you happen to have one. These were, it looks like, mechanically taken out of the animal. So a part of your pericardial sacs are be in different levels of state of existence. But I can find at least a part of it right here, right? This nice thin serous membrane. You can see how thin it is. Once you've identified the pericardial sac, look at it, realize that it fuses to all of the bases of all the vesicles going in and out. It seals around each and every one of them. So liquid can't get out of that chamber, that pericardial cavity. And I want you to see how durable it is. So if you have a little portion of it, like this little piece right here, right there, I can see through it. You can actually see the blue of my glove. But then just tug on a little bit and realize, damn, that is tough. Right? That's why if you get hypertrophy of the heart and it swells up, there's not a whole lot of room for it to swell up. Right? So you don't want it to do that. Look around, get your bearings. See if you can then identify the idea of the left auricle for the left atrium, anterior to ventricular artery, and that's where my great cardiac vein would be located also. Some veins you can see and some veins you're not going to be able to see, right? But see which ones you can and cannot see. Then we got to open them up. So I'm going to orient myself to look at the anterior part of the heart. I'm going to take my scissors. Don't need a scalpel for this. I'm going to take my scissors. And usually for some reason the left is always easier to access first. I'm going to rotate it sideways. I'm going to cut right down the middle of this auricle and into the atrium and take my scissors and just cut straight all the way down to about there. So I'm just going to take my good scissors, right? I'm not going to do it this one. I don't want to take someone's fun away. But I'm just going to lay it on its side, poke the auricle, get into the atrium, and then just cut to create like a little change purse. Just pop it open, I can look inside, I can see the ventricles, I can see the atrium, I can see the valves, the choroidal tendinate, papillary muscles, and then I'll do the same thing to the other side. All right, rotate it over. So this is the side that we first did. I'm gonna rotate it over, right? I can see my auricle, my atrium is right in there. Kind of look for the middle of the heart, stick your scissors in, cut, make the change purse, look inside. Ventricles, atria, you can tell if the left side or right side. Then I'm going to go around group to group, make sure you're doing that properly, but also once we get that done, you can find your left and right atrioventricular valves, otherwise known as tricuspid and bicuspid. You can see the choroidal tendinate. Pick the heart up by the cord, zzz, and bounce, just a light little jiggle. See how durable and tough those things are, right? Then find the superior and inferior vena cava. All right, your first task is, first you know intellectually where they go. Yes, I do. Then go there and start looking for them. I want you to notice the thickness of the wall of the vena cavas compared to the pulmonary trunk, compared to the aorta. Right? I listed them from the lowest possible pressure system to the greatest pressure system. That means your superior and inferior vena cava, you can almost see the color of your glove through the tissue wall. It's very, very thin. Pulmonary trunk has more pressure than the vena cavas do. And the aorta is about 2 millimeters in thickness. Right? That's really thick. Right? That's a high pressure system. Right? That's what we're talking about cardiac output today, in the 120 over 180. That's why it has to be so thick to resist so much pressure. Then I will go around, and for each of you, I will open up the heart for you to find the semilunar valves. So you can literally see them. And as we're looking at them, picture a kangaroo pouch in your mind. Now just picture a triangle of mother kangaroos facing each other, little bellies pointing toward each other, and the pouches that would be facing each other. That's what your semilunar valves are doing inside. Right? So you need to find all the chambers, find all the valves, notice the choroidal tendinate to the papillary muscles, find the superior and inferior vena cavas, and whatever else you can identify on here. Some of you have more or less of an aorta. You may not even have a brachiocephalic, or you might have one. Some of you have a 6-inch long, 7-inch long aorta, which is really cool. All right, some of you will have barely one at all. If you don't have at all any of the pericardial sac, right, find somebody like, this is a really long aorta, right there. right? Find someone that does, so you at least look at it. So maybe someone who is not that, who's a little more woozy with heart tissues and fluids, let them be the photojournalist. Right? If you can, take photographs of it before you cut, then while you cut, and then after you've cut. Because you will have these on your lab exam. 1, 2, or 3 of them. Right? And it shouldn't be a problem, because if you know your heart structures, whether it's a model or the real thing, you know it by geographical location of what it is. So it should be pretty straightforward. This should take you about an hour to do. Right? Once we're all done, cleanup time. You're going to take all your stuff, all your parts, and put it in an individual, small little Ziploc bag. Push all the air out, seal it shut. All right? Put your name or some kind of identifying group name, reference on that Ziploc bag. I will then refreeze them tonight so you can have them again on Wednesday. All right? So in case you didn't get a chance or you want to explore some new things or see somebody else's heart, we can also do it on Wednesday, because now they'll be pulled apart individually and they're faster to thaw. All right? This was one big large mass when we first got them. You will rinse off the trays. You will clean the trays off, and a little warning apparently. Number 1, you're also welcome to use gloves as many times as you want. They're cheap. This is my 3rd set for this lab group already, right? So if you want to use your phone, take off your gloves, re-glove, that's fine. Cleaning though, this is what you want to spray down when you're done. You want to clean your tools with this, clean the tray with this. The tools will go back in their proper tray after you clean them with this and rinse them off. You squirt the tray down quite liberally, get some water, kind of rinse it off, then dump all the water off and rinse it out really nicely. Alternate the stacks so the air can flow in there. Squirt down the bench with this. Be nice and liberal. Paper towels are cheap. Get it nice and clean. Don't try and clean your table as 2 groups did earlier today with this. This is cat preservative solution. It leaves the table really oily. And this poor student had her notes laying all over. I thought, oh, she's going to get her notes wet, but they'll dry off, no big deal. I came back 10 minutes later going, the table should be dry by now. What did you do? And I just kind of went, yep, wrong thing. Nice and greasy. All right, so don't use this. This is to keep cat tissue from drying out, right? Use one that says vinegar and distilled water or something, just bench cleaner, okay? Then we'll have access to them on Wednesday. When you're done with that, you've all cleaned up, you are adults, do what you need to do. I would stick around and work on my heart anatomy, my blood vessel anatomy relationships, but I also realize for you guys that haven't had the years of experience of running on vapors like I have, you may be tired, you need to go home, right? Have a few shots of tea, right? So I'll leave that up to you guys, okay? All right, come get your stuff and get started. I hope you guys that need help